Welcome to These Kids Can't Wait, discovering the world of rare disease. Hello, and welcome to our podcast today, where we explore the cutting edge of research, innovation, and the incredible stories of those making a difference in science and medicine. My name is Erin Hubbard, along with my co-host Laurel Gregier. Today, we're diving into the fascinating world of rare disease research, specifically Beta-Mannosidosis, a genetic disorder that is as rare as it is complex.
Our guest today is Dr. Angela Martin, a postdoctoral researcher at the Kimonis Laboratory. Her main interest is in inborn errors of metabolism, and her research focuses on lysosomal disorders, Pompe disease, and beta-mannosidosis. Her work on Pompe disease includes therapeutic approaches based on using antisense oligonucleotides, and her work on beta-mannosidosis includes a natural history study studying the biochemical and molecular phenotypes and potential treatments for the disease. She obtained her master's degree and medical degree in human genetics at the National University of Columbia. Outside the lab, she enjoys cycling, crocheting and embroidery, watching movies, reading, and supporting Columbia's national soccer team. Dr. Martin is fundamental to Kimonis Lab and the Lost Enzyme Project. We are so grateful for all the hard work she does to give hope to those affected with beta-mannosidosis and their families. Dr. Martin, welcome to the show.
Yeah, thank you. Thank you very much for inviting me.
Yeah, we're glad you're here. Dr. Martin, let's start by having you tell us a little bit more about yourself and your role at Kimonis Labs.
Thank you. Well, I am a postdoctoral scholar, so it means that I am devoted to research. I help Dr. Kimonis to design what kind of experiments we're going to do in turn to solve questions around beta-mannosidosis. And also I do the experiments, the actual experiments, and I analyze the results, and then I publish those results in a journal that goes to the people who are interested into the topics that I am looking for answers at our laboratory.
Great. And for those who may not be familiar, can you tell us what beta-mannosidosis is and how it affects patients?
Yeah. Okay. Let's first think about a cell that works as a big industry. When you need when you produce molecules, substances, you need to direct those substances to a specific place. And as when you send a mail package, you need to label in order to that package to reach the corresponding place. You don't want your Christmas package to go everywhere. So you need to label that. Once the package has reached its destiny, that label or that box, that envelope, you fold that and you trash to the recycle bin, and then it goes to recycle. Okay, that's what happens. In the cell happens exactly the same. So every single molecule that needs to go to a specific place into the cell or to the body needs to be tagged. And those taggings are made of sugar. It's similar to the sugar that we eat in the diet, but they are very complex sugars. So they are like tridimensional structures, completely different to what we eat. Those tags, once the molecule that is tagged reaches its destination, that tag goes into a place where it is recycled. What happens if that recycle is not able to be done? So those tags made of sugar tend to accumulate. So that is what happens in beta-mannosidosis. The defect in beta-mannosidosis is an enzyme or a substance that breaks those taggings, those sugar taggings, into simple molecules, into simple sugars to be used by the body again. In beta-mannosidosis, the enzyme is called beta-mannosidosis, and the problem is that this enzyme doesn't work. So what it happens is that these tagines accumulate into a place of the cell that is called the lysosome. And once the lysosome is not able to keep those sugars inside, they go out to the cell, tends to accumulate around the cell, and then it should escape somewhere. And what happens? It tends to escape through the urine. So in people with beta-mannosidosis, the way that they are detected, and in other people with similar conditions that are called oligosaccharidosis, the way that they are detected is through the urine, because you can detect all those excess of sugars into the urine. That is more or less what it happens.
So I'm curious, Angela, these sugars, um, do they all come out in the urine or are they toxic? Do they remain toxic in the body?
Most of them are in the body, in all the cells. But since they are not able to be degraded by the beta-mannosidosis, they accumulate, and that excess goes through where it can be excreted. So the main excretion is through the urine, but they are accumulating all the time, all the time, all the time. But at the same time, they are being excreted in a very high concentrations through the urine.
Okay. And what are some of the symptoms that these patients experience?
Mm, Actually, we found that in the lab, we found that the main manifestations of these patients are neurological manifestations. There are other disorders similar to beta-mannosidosis, like the oligosaccharidosis that I told you before. For example, alpha-mannosidosis, sialidosis, Kanzaki disease, Schindler disease. Those have different manifestations. The one that is more like knowledgeable to other people is alpha-mannosidosis, and you can recognize immediately those patients because they have physical features, they have skeletal problems. But in beta-mannosidosis, the main manifestations are neurological issues. That is, behavioral problems, including autism, seizures, hearing loss is the main symptom that they can manifest. It is the first symptom that is usually seen in these patients. Tags in the skin called angiokeratomas, that are little red spots that appear, and they are like elevated through the skin, and they can appear everywhere. And the other more advanced symptoms that we have seen is ataxia, progressive ataxia, some type of neuroregression, that is the kid that learns to walk, to write, to listen. They tend to regress all those symptoms that we have seen so far. And also we have seen that the physical features are very subtle, and you can see that, hey, but he or she looks like a normal kid. They don't have like weird things on her face or his face. Yeah, that's what we have noticed in beta-mannosidosis.
Mhm. Yeah, and just from personal experience, I can tell you this is incredibly challenging to see these symptoms, you know, in these patients, and it's hard for the families. How common is beta mannosidosis?
Is extremely rare. We call it an ultra rare disorder because it's very, very rare. And the places that have published uh, incidence of beta-mannosidosis is because they have a population with the same uh, type of mutation of genetic variant. For example, Czech Republic, Slovakia, Hungary, because they have a population there that have beta-mannosidosis, but it is a special type of population, that it is the Roma population. They are gypsies. So it is a very specific people that live there. But in other places is very rare. We can see that it's 0.16 per 100,000 living living birds.
And I'm just curious, Angela, I mean, with it being so rare, do you think that it is so rare because it's under tested for and under identified? I mean, are there things that kind of also contribute to its rareness?
Mm, Probably what we have seen in what we have researched at the lab is that we do not rule out that it could be underdiagnosed because these patients have a spectrum of symptoms where they can be very mild, that you can notice, oh, maybe he has problems with the hearing loss, and you put the hearing aids, and that's it. Or you can have the severe spectrum, where they have very severe symptoms, severe seizures, ataxia, they cannot walk. But probably there would be a high amount of people that are in the mild spectrum, that probably they have only the hearing loss and some spots on the skin, and it doesn't look like a very rare disease. So we cannot rule out. We need to do more research on that, but probably that's the reason why, one of the reasons why it is very rare.
Is this the most rare disease that you've worked on?
Yes, definitely Beta-Mannosidosis is the rarest disease I ever worked with. Even my professors haven't seen one patient with that.
I'm just curious, with it being so rare, is that intimidating or is it more motivating to you?
It's extremely motivating because you now you have a purpose to find if it is really rare as it seems, or if these patients are being underdiagnosed. Yeah, because the best thing that could happen is that these patients are being underdiagnosed, so it means that we need to work harder to find these patients and to start looking for treatment.
Yeah, absolutely. And we're excited to help you with that too.
Before we move into the next section, Angela, I just wanted to say I really liked your analogy, kind of simplifying that explanation of what is beta-mannosidosis, how does it affect the body. As you were talking, I was just envisioning my home receiving all of these packages. You know, my home is the cell essentially, and all of these packages with no place to put them, building up until it looked like, you know, an episode of Hoarders, and packages to the ceiling, packages out the window, across the lawn. And you know, when you have that visual, you can really start to understand how devastating that is to the body. And when you think that's happening in all the cells, you know, it just really emphasizes the need for treatment, to improve that. So let's talk about your work on the enzyme replacement therapy for beta-mannosidosis. And maybe when you're talking about that, you could explain how the therapy works and what the J-brain cargo technology is and why that makes it such a promising approach to treating beta-mannosidosis.
Since there is a problem with the enzyme, yeah, maybe you can think, okay, why don't give in the enzyme? Okay, with lack of the enzyme, let's give it the enzyme. Okay, it's not that easy because the brain has a single feature, and the brain, it is protected by a barrier called the brain-blood barrier that only allows certain substances to enter into the brain. And it is amazing because it protects our brain from a lot of substances that we have in our bloodstream that could enter and damage the brain. So it's really necessary, and it's an amazing structure. But at the same time, it is a huge problem because the enzymes that are huge molecules that are made of proteins, those substances are extremely big. It's trying to put an egg through the hole of a needle, you know? And it is huge for the brain blood barrier, and it won't cross. Okay. So the development of enzymes in every research are trying to bypass that barrier. And the J-Brain cargo is a technology that was built with that purpose. That means that the enzyme that we want to enter into the brain will be attached to a specific molecule that it is recognized by the brain blood barrier, and then the brain blood barrier will allow that molecule to enter. So it is like a gate, like a portal. So this portal will open the pathway to the enzyme to enter into the brain. That's how it works. Without that special molecule that it is the J-brain cargo specifically attached to the enzyme, the molecule won't be able to enter. I mean, the naked enzyme won't cross the brain blood barrier. But if it is attached to that J-brain cargo, the J-brain cargo will open the gate to enter into the brain. Okay, and it is amazing because it bypassed the main barrier that allows us to develop to deliver the enzyme into the specific place that we need to go, that it is the brain, taking into account that the main manifestation of beta-mannosidosis are neurological issues. So this is amazing. And the great thing is that this molecule has been already approved. I mean, this technology has been already approved as a treatment for other similar conditions, similar lysosomal storage disorder, as mucopolysaccharidosis type 2 in Japan. And the clinical research that has been done with this molecule are amazing. So I believe that this technology is great. So we were very lucky to have JCR, the company that is helping us to develop, that are developing this enzyme, and we are very honored to work with them, and it's awesome.
Yeah, I was going to say for our listeners out there, so JCR Pharmaceuticals is the biotech company that has engineered this amazing technology. And so, it's patented, it is something that they've created, and it's just a complete game changer. So we are so honored and just incredibly blessed to have JCR, come on board for the Lost Enzyme Project. So, Angela, you got to spend time over in Japan with JCR Pharmaceuticals. Can you tell us a little bit more about that experience?
Sure. I can say that it has been the greatest experience I had in 2024. And not only from the standpoint of a scientist, but also as a person. That means going to Japan was always the dream of my life. For everyone that likes anime, it is the dream to go to Japan. But it was a great experience because I didn't taste the flavor of Japan as a tourist, but as a scientist. So meeting the people at JCR was a great experience in terms of I learned a lot of them, how well they are organized, how great the teamwork is, and how warmly they received me, and how eager they were on teaching me all these technologies, all these experiments that I need to replicate here at the US. So, and all the support that I received from them. So I really felt very honored, very well received, and I learned a lot, not only from the science and laboratory, but also how great people are Japanese people. They are great to work with. And hopefully we keep continue doing the great job for this, especially for the patients.
Is there one memory that stands out kind of above the rest of your time there?
Yes, there's a lot of memories. Some of them were that um, they are learning English. Some of the workers are learning English, So most of the time I was speaking English, although I'm not. It is not my first tongue. But it was an amazing time because we were learning, both of us, something different from the laboratory. And also I tried for the first time, I tried frog meat.
Oh my goodness.
It tastes like chicken, so it's delicious. I recommend it.
So, Angela, I'm really curious now. You got to go over to Japan. You had this wonderful experience. Do you remain in contact with the team over there, and kind of how does that impact the phases of this project? And maybe you could also tell us, what the general phases of this project are, and kind of where you are in all of that.
Oh, yes. I have received total support from them. That is an extremely important part of this project, because basically they are doing half of the project. We are doing the other half. They are developing the molecule, making uh, it safe for use for humans. Yeah, and also they are developing the experiments to be applied on mice as well, because there are different steps to be tried first in humans, then mice or cells. There are different steps that we need to reach. So they are doing all this hard work and synthesizing an extremely complex molecule. It's not an easy task. You have a lot of hindrances, you have a lot of difficulties over the path. Uh, and finally, they did it, and it's amazing how they did, and it's such fast pace. That's one thing, and the other thing is that we cannot work without them in terms of the lab is doing all the translational studies, but the molecule behavior are known by them better than us. So we work together in order to make this project thrive. And
That's really
that's
amazing.
that's one thing that I have received all the time. I have in contact with them all the time. I receive mails, I send mails, hey, I need help with this. Yeah, do this, do that. So it's also a great support that I have had from them.
Yeah, yeah. To have a strong partner like that is just incredible. So where are you right now? Like, if you could maybe explain to us a little bit about the phases of the project, and then where you are right now in those phases.
Yes, the first part of the project is the basically well design. Once the molecule has been designed, what we need to test is if it reaches the place that we want to go. So we need to prove if the molecule reaches the brain tissue and the neurological tissue. We already have tested that, and yes, it goes to the brain. So that's the first part. That's great. Now we need to develop a test that is called the pharmacokinetics and pharmacodynamics. That it means how the drug will be distributed into the body. Okay, because it reached the brain, but how much of the drug goes to the brain? Because we need to a certain amount of drug that to go to the brain in order to have the effects that we are looking for. That is the reduction of the accumulation of the oligosaccharides in the brain. Okay, so that is the second part. We need to design how the drug will distribute to the body. Once we know how it is, then we can calculate how much drug we are going to give to for treatment. That means I am going to try. Now I know how much of the drug every single dose will have. Then the following part is to test that amount of drug in two models. The first model is the cell model, because it is in vitro. We test the cells from affected patients and see if they recovered or they improve, or if there is effect or not. Okay, and At the same time, we try this molecule in our second model, that is the mouse model. We are going to give the drug to this mouse in order to see if this mouse improve. This mouse has the condition, has the disease. So what we are going to see is if we see a reduction of the accumulation of oligosaccharides in the tissues, but we also want to see if there are changes in their behavior or in their coordination or in any other neurological features.
So, Angela, once, let's say you see these changes, these improvements, how long before you can start testing this treatment in humans?
We are specifically in cells, we can see almost we expect to see them in 24 hours to 48 hours. That's what we expect because we are delivering the drug to the cell directly. In mice, we don't know because we don't know how long it takes to the drug to make a visible effect, to have an effect that we are able to look after. We can see very microscopical changes, but we don't know how long it would take. Okay, so that is also a part of our studies. Once we treat the mice, we can check at what time does it take to reduce uh, all the burden of the accumulation of the oligosaccharides. So that's part of the design of the project. Okay, we can see that it works better in very small mice or maybe in older mice. We need to see along with the project. We would be able to see how long does it take to the drug to make effect.
That sounds like a really tedious process. When will you start testing the mice? Have you already done it? Or is that something you guys are going to be doing soon?
We are currently testing the mice. This mouse model was published 20 years ago. And what it was published is that yes, this mouse have similar changes in tissue as human have. However, in terms of behavioral abnormalities, cognitive problems, hearing problems, or even coordination, they said that they were normal. They were not big differences. So what we are doing right now is doing all the natural history of this mouse model to check if they are really having any problems that at the time of publication weren't discovered. So we are looking after coordination problems. So we are doing motor testing on these mice. Also, we are looking after learning problems, hearing problems, and cognitive problems in these mice. So we are doing a cognitive testing as well. And once we got these results, we will have a baseline of knowing, okay, does these mice, do these mice have problems as humans have or not? We haven't encountered that some mouse models have very bad problems in the tissue, but nothing outside. And it frequently happens. I am pretty sure that these mice have problems, but I need to discover them. So we are currently undergoing that project, that part of the project.
And that takes time.
It takes time, yes, because since we don't have any data, we need to check how long it will take to the disease to make physical manifestations. Okay? And so it means that the mouse will be tested until they are 1 year, 2 years, maybe earlier if the manifestations are early, or maybe longer. Yeah, all the lifespan of the mouse that it is around 2 years and a half, 3 years.
How long do you need to observe them and test them before you can move on to clinical trials with the children?
The behavioral and cognitive and coordination problems in the mice should be observed, and we are already observing for the lifespan of the mouse. Okay, but we are doing many things at the same time. It's not like I am doing this, then I am doing this. No, we do multiple things at the same time, and based on those results, we can do other multiple things at the same time. So while I am looking over the lifespan of the mouse, how they behave, how they coordinate, everything about their phenotype, at the same time, I will be working with the cells. Yeah, because the cells have a big amount of time to be worked with. Once we have the results, probably I will have the results of the mouse. So now I have a baseline and start the pharmacokinetics and pharmacodynamics study. These pharmacokinetics and pharmacodynamic studies last at least between 3 and 6 months to have it completed. And once we have that, now we can start doing clinical studies with the mouse. And it takes a year, more or less. Once we have those results and we are sure that it's safe and we are sure that it improves the phenotypical features in animals, then we can proceed with humans. Because we need to be sure that everything is going well before giving something that it doesn't work to humans. Does
Absolutely.
it make sense?
Mm-hmm.
Yeah. The good thing about this is that the molecule itself, like the J-brain cargo, is not a new, well, relatively is new, but it's not like a very, very new technology. It's not the first time that will be used. There are at least 3 conditions that have used this technology for enzyme replacement therapy. That is one thing that it encourages us to keep working. The other thing is that we are not only working with mouse model, we are working with cells that are derived from humans. So it is like a very small representation of what we expect to have in humans. So we are already trying this molecule in human tissue, and we are having pretty good results. So we are doing everything, and we want to be sure that everything will be ready to be applied in humans because we want to give them the safest and best treatment.
Yeah. What are some of the biggest challenges so far that you're finding reaching that goal?
Well, the biggest challenge is the small amount of patients. It's since it is a very rare disease, not many people know about this condition, and in consequence, many people don't have that differential diagnosis in the back on their minds, sorry. So what you don't know, you can't see. So if you don't know about this condition, or at least have heard about that, it's pretty difficult that you have into your differential diagnosis as a doctor. And it's completely normal. It's expected because the people who treat rare diseases are geneticists. Okay. But if the neurologist and the geneticist would know about this condition, probably the amount of patients with beta-mannosidosis would be higher. So I believe that this is the first thing that we need to do. It's to educate the people about this condition, to tell them, hey, this exists. And probably it is underdiagnosed because there are very mild patients that would have it, or other patients that you believe that have this, but in fact that they have beta-mannosidosis. So it is completely different
Absolutely.
things.
You know, I've noticed one of the biggest discrepancies really in literature, until recently, is just this lack of recognizing, the white matter changes and white matter issues in humans. In previous literature, it was always like, oh, it's more mild in humans, and, we're not seeing any white matter changes or white matter issues, myelin issues. Can you maybe talk just a little bit about that?
Yes.
And why neurology is so important as a puzzle piece of like, hey, this is a diagnosis here.
Yes, part of our work at the Kimonis Laboratory is trying to figure out the natural history of the patients. That means how the disease goes over time, how the disease behaves over time. So it is important because knowing the natural history would make easier to the physicians to recognize how the disease can be found in certain times of life of the patient. Yeah. If it is a newborn patient, or if it is a patient with 10 years, or a patient with 30 years, if you know the natural history, you would be able to recognize the disease in this patient. Unfortunately, with beta-mannosidosis, there is not a natural history that has been published before. So that's what we are working at. What we found is that the most common symptom is hearing loss. And hearing loss is extremely common in population because anything could cause hearing loss. Okay? So that's another point that makes us think that this condition is underdiagnosed. Okay?
But now not everyone will have hearing loss with beta-mannosidosis. Okay.
Yes. Hearing loss is extremely common. So that is one point. Another point is also we went over all the patients that were previously published, and we found that there is a specific change in brain MRI that compromises the white matter, and it is the lack of myelin. Myelin is like an oily substance that covers an extremity of like a limb of a neuron. So that limb that it's called the axon makes the neuron to communicate with other neurons or to communicate with other organs, for example, muscle. So when these neurons, to make that communication faster, they are surrounded by this oily substance that is called myelin. So what would happen if this neuron doesn't have myelin? First, the communication will be slow, and second, the axon itself can be easily destroyed because the myelin also protects these axons. So what we are seeing right now is that the common feature in the brain MRI of these patients is lack of myelin in white matter. And it is extremely interesting because it seems that although not all the patients have this feature, having hypomyelination allows us to think immediately that first, there should be a genetic condition. Second, probably there is lysosomal storage disorder. Third, I need to take into consideration beta-mannosidosis.
Absolutely. I mean, that was how Oliver was diagnosed. You know, he was one of the rarer ones that didn't present with any hearing loss, and his hearing today is great, but it was the myelin, you know, at the neurologist. His MRI was abnormal, and exactly what you said happened, and that was how he was diagnosed at the Mayo Clinic.
This concludes part 1 of our interview with Dr. Angela Martin. So much ground was covered already, and we still have much more to come. In part 2, we pick up right where we left off, diving into the real-world impact of her research, the role of family advocacy, and what gives her hope for the future. So join us for part 2.
Thank you for joining us on These Kids Can't Wait. Every story shared brings us one step closer to awareness, advocacy, and action. If you've been inspired, please subscribe, leave a review, and share this episode.
To learn more about the Lost Enzyme Project or donate to the cause, you can visit our website, Instagram, or Facebook page. Those links will be provided in the show notes below. Thank you again, and remember, these kids can't wait, but together, we can make a difference.